Screening by PCR. We discussed previously that PCR can be used for the screening of different DNA from different sources. Although it can also be used to isolate our gene of interest from the source DNA by the process of amplification, or you can say that uncloned DNA can be used for the isolation of specific gene by PCR. But in addition to that, this PCR, it can also be used for the screening of DNA libraries or cDNA libraries. For the screening purpose, we have the idea what may be the prerequisite or the condition that we must have the idea about the target gene, that basic information, if available related to its sequence, then primers can be constructed, and these primers can flank the target sequence so that its amplification it is there. So in this way we can also detect particular gene from any source, either from the genomic DNA or different type of DNA libraries. Like the DNA hybridization technique, PCR it is also very, very important and can be performed with accuracy. For example, in case of DNA hybridization, we need the growth of microorganisms, their lysis, and after lysis, the immobilization of DNA on nitrocellulose membranes, and then their detection by using probe, and then finally autoradiography can be used for detection. So all these steps can be avoided by using PCR if we have the idea of the primers. So these are the primers for that particular target gene. Then we can perform the PCR study. So here multiplate PCR it can be used. That plate which is commonly used for PCR that contain multi wells. So the cloned DNA it can be dispersed in different wells of the PCR plate, and then the specific primer they are used, and the wells that contain the amplified product, we can say that this contain our target DNA, and then with the passage of time it is further diluted, and again PCR reaction is performed so that we can detect the accurate target DNA. And in some cases like the degenerate probes, degenerate primers, they can also be used if we don't have the idea about the sequence of the gene. Rather, the sequence it can be deduced or decoded from the sequence of the amino acid. So in this case, if the primers they have been constructed from the sequence of the amino acids, तो इस सिचुएशन में जो प्राइमर्स हमारे पास आएंगे उनको कहते हैं डीजेनरेट प्राइमर्स. डीजेनरेट प्राइमर्स एक्चुअली ये मिक्सचर होते हैं प्राइमर्स का जिनका सीक्वेंस मोर और लेस सेम होते हैं, स्वाय चंद एक बेस पेयर्स पे वेरिएशन होती हैं. तो इस तरह से लाइक द हाइब्रिडाइजेशन प्रोब वी कैन आल्सो डेवलप degenerate primers by keeping in view the sequence of the amino acids. While constructing the degenerate primers, again we must also have in our mind about the degeneracy of the different amino acids. And again here those amino acids like tryptophan and methionine that are coded by one codon, they can also be incorporated so that low degeneracy may also be incorporated. So in this way the primers can be constructed if we have the idea about the sequence of the target gene, or in case if we have the idea about the expression product, if we know the sequence of the amino acids, degenerate primer can be also used for the screening of the DNA libraries.